Okay, thanks for your patience there. So if after the session today, you do want to meet with any of the companies that are presenting, we will share, facilitate the sharing of those details. And, that's between you and the supplier on how you follow up, if you do that under secrecy or not. We do, as GC3, we remain available if there's any assistance needed. And the whole concept of Shark Tank was the possibility that there might be the need for a one-to-many kind of relationship instead of the traditional one-to-one that you have, and as a supplier to a downstream partner or a customer. If we do need to be involved in that, we're happy to do that. But I think initially, today there's going to be a lot of questions, I'm sure, about the technology. Before you start getting into the collaboration, people want to understand, is this going to meet your needs? And that makes a lot of sense. We want to remind people, we don't have any secrecy agreements in place today. So, we've asked USDA not to share proprietary information. And please keep that in mind as you ask questions. It's always possible they may tell you that, I'm sorry, I can't answer that question without a secrecy agreement. So because we do have potential competitors in this session, I do need to show and read part of this antitrust statement to you. First and foremost, I just want to make sure it's clear, GC3 is, we're not here to play any role in your competitive decisions of whether, of our members. We're not here to restrict competition. Honestly, our goal is to promote the commercialization of all green chemistries, and that is our intent. So, but we do know, as potential competitors, I just want to remind everybody, please, keep in mind, we can't be talking about anything that could be an antitrust concern around any of the laws around the world. This could include we got one more folks going, so we can't have any discussions regarding individual pricing or marketing plans, or terms and conditions, sale negotiations, changes in capacity. We can't be talking about market share or customer allocation, company boycotts, no discussions around refusals to deal with people or any specific matters that would be of an antitrust concern, such as, bidding. So any of those topics, just a friendly reminder, those need to be things that we don't talk about today. So with that, I'd like to turn this over. We got a really interesting presentation from USDA, and we've got quite an audience. So thanks for joining us today. I'll come back in at the end after Billy gets done presenting. And Billy, I'll let you go ahead and please introduce yourself as well as your the team from USDA.
Great, thanks, Kevin. I'm Billy Hart-Cooper. I'm really excited to be here. I'm presenting on behalf of our group, the Bioproducts Unit. We're a research unit within the Agricultural Research Service. Jim McManus is here, he's on our team, and Bill Orts is our research leader, and he might make an appearance. So I'm super excited to be here today. I participated in the GC3 Preservative project a number of years ago, and some of the feedback that I got from all the participants was extremely helpful in future directions on this project. So I'm really excited to hear what you think. Any constructive criticism and feedback would be super helpful. So I'm going to talk about our novel preservative solution and disinfectant and wound healing properties of that material. And just as a little bit of background, I started on this project over 5 years ago, working with Method to investigate natural materials and non-toxic synthetic materials that could be useful preservatives or alternatives to conventional preservatives. So long story short, I screened hundreds of different compounds against organisms of interest, Pseudomonas aeruginosa and Aspergillus brasiliensis. And from all that data and results, some patterns emerged which suggested that certain chemical functional groups are highly effective as antimicrobials. But one issue that we kept coming back to is the same chemical functionality that makes it an effective antimicrobial also provides and causes hazard, both hazard to humans and the natural environment. So for example, quaternary ammonium compounds, highly effective antimicrobials, functionally pretty simple. It's a positive charge and it's a hydrophobic component that are joined together in the same molecule. But they can often be persistent, especially under anaerobic aqueous conditions. And when exposed to bacteria in the natural environment, they can produce antibiotic resistance to clinically important antibiotics. Now that's a big interest for USDA because antibiotics are used in agriculture. Rising antibiotic resistance is a big problem. It's been estimated that in 2019, there were 1.2 million deaths worldwide that can be attributed to antibiotic resistance in bacteria. And that number is expected to increase about an order of magnitude to about 10 million deaths per year by 2050. So the main takeaway here is that we were very interested in trying to identify new strategies to separate performance from hazard. Both performance and hazard are related to chemical structure. So can we get performance while reducing hazard by creating chemicals that change structure over the course of their lifetime, going from hazardous to non-hazardous? And so the scheme we devised, oh, I should share slides, maybe that's better. The scheme we devised was to identify 2 non-toxic, very low hazard ingredients that have a history of use in consumer products and are ideally naturally sourced. When they're exposed to one another in a formula or before you add them to a formula, they form an active reversible complex that's shown as C, which is very potent, very strong antimicrobial, but also non-hazardous in the ways that you want it to be, like non-toxic to skin. Then once you're done using the material, you spray down your surface, you wash it down, the residue goes down the drain. Water will drive it apart. It will dissociate passively as it's diluted back to its subcomponents. So these subcomponents are, 3,000 times less hazardous to organisms and even microbes. Effectively, they're not antimicrobials anymore. So there's low risk of developing antibiotic resistance compared to a compound that doesn't dissociate. And these compounds are also pre-selected so that they biodegrade fairly rapidly. So how does this actually work? Well, we screened hundreds of different compounds again, threw spaghetti at the wall like we do, and identified a combination that's composed of cumin aldehyde, which is the active flavor in cumin seed oil, tastes like tacos, smells like tacos, and aminoguanidine. Now, this is a small molecule that has extensive human testing, has been shown to be biodegradable under aqueous conditions. It was investigated as a treatment for diabetic nephropathy, and it's gone through phase 2 clinical trials before it was abandoned for lack of statistical significance. So human testing has been done. Close to 700 people have consumed, anywhere from 300 to 600 milligrams of this stuff every day for 2 to 4 years, with either reversible side effects for the high dose group or no side effects. Now, when these 2 compounds are subjected to each other in an aqueous solution, they form a fairly strong, fairly stable, but also reversible bond, this hydrazone bond here. And the resulting compound is non-irritating and non-sensitizing to skin, according to our repeat insult patch testing, and also non-volatile, less likely to be inhaled. So how does this stuff actually work? Well, we identified a base formula that, without any sort of preservative, grows microbes very readily. This is a non-ionic spray cleaner, similar to what you might put a quat into. You can see on the top graph that it tends to grow most microbes fairly readily. It didn't grow staph. When you add just 0.2 weight percent, so 0.2% of our active ingredient, it knocks everything down. And it keeps it that way. We've done it extended USP 51 preservative challenge testing and shown that these levels are stable. And even after you re-inoculate the formula with, say, aspergillus, which is the hardest to kill organisms that we've observed in this test, you still repeatedly knock down that aspergillus by at least 2 logs and maintain passing criteria. And this was done over 90 days. Jim did these experiments. We became interested, based on feedback from the last GC3 round, in the disinfectant properties of these compounds. And so we did some homemade tile disinfectant tests, and we observed in some cases that our compound performs near identically to benzalkonium chloride and bleach at 0.05 weight percent, so keeping the same concentration for all those. So our compound is the green dotted line in the top graph, benzalkonium chloride is the red dotted line, bleach gave the same result, and then our control, which is no chemical added, just water, is the blue line. From that feedback, we went back and did some drop disinfectant tests and suspension time kill tests, and also looked into some secondary benefits of these compounds. So I'm going to summarize. I can talk more about this if there's interest, but in terms of the drop disinfectant tests, we saw very high performance against gram-negative bacteria, Salmonella and Pseudomonas. We saw slower kill kinetics against Staph aureus. So then we became interested in adding boosters to see if we could enhance the activity of against Staph aureus. So we tested it in different non-ionic spray cleaners. Adding our ingredient to those spray cleaners enhanced the performance. So we saw up to 4 log reductions of Staph aureus within 10 minutes. We also saw that we got enhanced formula compatibility by adding those boosters. And it became the subject of a second patent application. As many of you know, quats have secondary benefits as surfactants. And so we examined the surfactant properties of a series of different derivatives of this basic structure and observed that you can obtain a wide range of different critical micelle concentrations, foaming abilities, and oil water emulsification properties, depending on which derivative of this compound you're testing. I'm going to go over this real quick because I know it isn't a major interest of the group, but it's really, really exciting to us. And so I wanted to share it. There's a really gnarly and horrible hoof disease called digital dermatitis that's very common in hoofed animals, both domestic and wild, cattle, sheep, even wild elk. So once you have this disease present in your feedlot, for example, or your farm, it can spread to wild populations. Now, once animals get this disease, they develop these painful polybacterial ulcers on their feet. I'm choosing not to include photos of it because it's pretty gross. And they get so bad and they're so hard to treat that the animals stop walking, they stop eating. And in order to cure the animals, it costs the farmers hundreds of dollars, up to $500 a head of cattle. And one of the big issues is that farmers can't use antibiotics to treat these animals because antibiotic residues are tested in milk and meat. So there really aren't any good treatments for this. If you are a typical sheep that has this disease, odds are it'll get worse. About a quarter of the sheep show no improvement, and then a quarter show some improvement. So testing our solution here, our collaborators applied it as an anhydrous balm, and we observed that within 1 to 2 weeks, all of the hoof infections were cured. Now, this result was basically identical to applying pure tetracycline to the wound, which is the gold standard in treatment, which is great for research and for animals that aren't part of the food supply, but you can't use that solution for dairy and meat producing animals. And so what we've developed here is really unique in that way. There's nothing else that's being sold commercially like this. And we think it has huge potential, not only for animals, but for also for people. Now, one of the big reasons why we're interested in this, why we're doing this, is because we want to develop antimicrobials that are less hazardous. I'm going to go through this pretty quickly. I'm glad to answer questions as they come up or have later discussions. But there's a couple main advantages of our compound. In many ways, its hazard profile is similar to quats if you're just looking at the active complex. I'd say one advantage that we have over typical preservatives is the very low hazard to skin. We did repeat insult patch testing, over 50 participants at 5 to 10 times the use level, so up to 1 weight% in a non-ionic spray cleaner, which is a commercial product, and we observed 0 cases of sensitization or irritation. Now, as part of the sheep studies, we also did irritation tests there and observed that at very high levels, you do see some, but for the practical use level, irritation doesn't occur within an order of magnitude of what use level you're going to be using it at and what you want to be using it at. The primary, and of course, the major advantage of this type of chemistry versus traditional cationics like bisbiguanides or benzalkonium chloride is that the subcomponents, once the dissociation occurs, and that can take anywhere from a few days to a few months, depending on the conditions, it's not instantaneous. But once that dissociation occurs, the subcomponents are effectively non-toxic, not only to environmental organisms, things like aquatic crustaceans or fish, but also to bacteria. They're functionally different, and we don't expect them to cause the same resistance problem as typical persistent cationics have been shown to. So what's our value here? Well, we found that by making these compounds reversible, we can start to separate hazard from performance. And like I said before, the reversibility isn't instantaneous, but it's a step in the right direction, we believe. It takes anywhere from a few days to a few months, and it can vary depending on the environmental conditions. But we're studying that and we're getting a better understanding. The subcomponents are highly biodegradable. Aminoguanidine biodegrades with a half-life of around 30 days under aqueous conditions. Human aldehyde degrades with a half-life of 1 to 2 weeks. The preparation of this compound is really an important strength of it. It's more akin to formulation than traditional chemical synthesis. You combine the two ingredients. It can happen in water. It can happen in a non-aqueous formula, but they self-assemble to form the active compound, and the only byproduct is water and a little bit of heat. So we've been able to make kilogram scale of this stuff on our bench without any issue, and it makes a highly concentrated, ready-to-use, flowable product. As I mentioned before, we've tested the ability for these compounds to interact with boosters and have observed that a number of commercially available preservative boosters can enhance the kill kinetics, make it more effective against gram-positive bacteria like Staph aureus. The compound is already extremely effective against almost every gram-negative we screened. And the combination with the booster enhances the formula compatibility of this type of chemistry. Now for the interesting part, we're not a company. As a government agency, we can't commercialize this ingredient on our own. And so our job is to work with industry and to help industry. So we're really interested in finding partners that can help produce and register this ingredient. But more broadly, we're interested in understanding where you're coming from and identifying how we can work together. What performance and applications are you seeking? Are you seeking registration? And if so, what type? You can always contact me or Jim. You can Google our names if you lose this slide. Contact us for samples and if you want to have further discussion on this. And I'd like to thank our research group, the Bioproducts Research Group in Albany, California, and our collaborators at Method SCJ.
Okay.
That's pretty much, yeah.
Great, thanks, Billy. Great presentation, thanks for that. Before we open it up to questions, I just want to offer a quick thought about, you know, as you look at the obstacle here, you know, as Billy said, as a government agency, they can't commercialize on their own. So they're seeking these partners to produce and register this ingredient. Obviously, this has financial implications. What I'm asking for today is please, any discussions about how you might partner, how you might, you know, what the financials might look like. That is something I'm going to ask you to do one-on-one with USDA after this meeting in a private meeting. For today, I want you to ask all the technical questions you want, ask about what they know about different, you know, this material and different applications, you know, those kind of things. We just need to avoid any of those financial kind of questions. Those should be done one-on-one, most likely under a secrecy agreement. So thanks for that. I'll remind everybody, when you ask a question, please let them know who you are, what company you work for, and make sure you unmute your phone. With that, we'll open up to questions.
Hi, I'm Jane Michelson from Target, and I had a question about, you mentioned throwing spaghetti at the wall early on in the presentation and that process of initially screening a bunch of different compounds. Could you tell me a little bit more about what that process entailed?
Sure. So we've thrown spaghetti at the wall multiple times. Initially, we screened a lot of different commercially available compounds to identify what functional groups were effective. And then for the reversibility strategy, we identified a lot of different compounds that had complementary functionalities that would allow them to self-assemble into an active compound that we thought might have some activity. And so, you know, one example of that would be screening a lot of different hydrozides, of which aminoguanidine is part of that class of compounds, which just means you have, you know, a hydrazine fragment attached to a carbonyl or imine fragment, like shown here. And then the second component would be either an aldehyde or a ketone, which could undergo condensation with a hydrazide to generate a fairly stable compound. The reason why we got interested in hydrozides, they have advantages and disadvantages, but they form this hydrazone that allows you to have a stable compound at ppm levels of dilution. So it's stable down to the level that clinical antibiotics are stable. And then below that, once you dilute it a couple orders of magnitude, it starts to break apart significantly. The dissociation constant describes a compound with stability down to ppm levels, and then it breaks down below that. So if you took like a liter of, I don't know, laundry detergent that had 1 weight % of this compound in it, and you dumped it in a 25-meter swimming pool, allowed it to reach thermodynamic equilibrium, then it would be over 90% dissociated based on the stability constants for this compound. So it's useful because it remains stable in aqueous solution down to pretty low levels, but it still dissociates. There are other types of imine bonds that are less stable and that would be less suitable for aqueous formulas. So that was our approach of throwing spaghetti at the wall. Combine a hydrazide with an aldehyde or ketone.
Awesome, thank you.
Hey, it's Chris Euchert, also with Target. I'm sorry if I missed it, but did you say what pH range this compound is effective in?
So we've screened a number of different pH ranges. Our understanding is that preservatives are needed in the pH 3 to 10 range. And so we evaluated all those different ranges. We generated some generic all-purpose cleaners, non-ionic spray cleaners that were pH controlled to be every pH within that range, 3, 4, 5, whatever, up to 10. And then we measured their performance against our hardest to kill organism, Aspergillus, and we observed similar performance across all of those pHs, although the kinetics tended to be a little faster at the higher pHs. But in all cases, we saw at least a 3 log reduction of Aspergillus within about a week. So
Nice.
broad pH compatibility.
Thank you.
Mm-hmm.
And just a friendly reminder, if you're trying to ask a question, you might be on mute.
Yes, hi. My name is Jason. I'm from Ashland Specialty Ingredients. I actually have a couple of questions. One of them is the raw material, humanaldehyde, doesn't have an antimicrobial activity on its own.
It does. Its minimum inhibitory concentration is about 1,000 times higher than the active compound. So if you were able, and solubility is a big limitation there because it's such a hydrophobic compound, but if you're able to solubilize it, you can get some antimicrobial activity. It's just at a very high level. And it's not reliable. It's not broad spectrum. Like it tends to not be effective against Aspergillus and Pseudomonas, but there are organisms that it's effective against. You just need a much higher level. So it's 3 orders of magnitude less effective than our active compound.
Now, a related question is that when you use your active compound in a cosmetic formulation, does it have an odor? I'm pretty sure humanaldehyde has a pretty strong odor.
Humanaldehyde does have an odor, and our active compound doesn't, as long as it's made in a pure way. So initially when we were making it, there was always some residual humanaldehyde in there. You can get around that by monitoring the reaction very carefully. Sometimes the easiest way to do that is to just waft it and smell if there's any residual humanaldehyde. There aren't many, the human nose is pretty sensitive. So if you make the active compound in the correct way, you let it run to completion, have a slight excess of aminoguanidine that would trap out all the available humanaldehyde, you can make it odorless. Now, because it's reversible though, like if you have it in a formula and a small amount gets reverted, then you might smell it.
Thanks.
All right, I'll just ask again, if there's any remaining questions, please make sure you're not on mute. We'll give everybody a minute here just to make sure there's no additional questions.
So, I just had a follow-up to that question too. The other thing too is it's not strictly human aldehyde that we're testing. It's kind of the focus of our presentation, but there are lots of other aldehydes that are odorless that you can use and give similar results. So in our original patent, we screened like 100 different compounds, and those are included in the patent. And human aldehyde is one of the few that has a very strong odor, but you can get similar performance from other ones that don't have an odor.
Thanks.
Mhm.
Okay, I'll give one last chance. Any last questions, anybody? Take yourself off mute. Okay, first of all, I want to say thank you to Billy for a great presentation today, and I want to thank all the participants as well. Just as a reminder about next steps, potential next steps. If you are interested in a meeting, with USDA, please let me know. I'll make the introduction. We certainly encourage as much follow-up as you want. We'll make that happen right away. And I know some of your colleagues from some of the companies could not be here today. This session is recorded, and that should be available by early next week. We'll have that available, and I'll communicate that to everybody so they know to look for that. So with that, I'll say thank you. I'm going to ask Billy and James to stay on the line, and thanks everybody else. You're free to log off at this point.
Thanks, everyone.
Thanks. Nice presentation.
Yeah, thank you. Thank you.
Thank you.
Thank you.